let's say, the reviewers from the review section. Now, the main difference here, if you notice, is something called as, let me see if I can open up my pointer. Yeah. So if you see, the main thing over here is this, the difference, peer reviewed and non-peer reviewed. So when I talk about journal, so today's topic is more about peer reviewed literature, because as Dr. Agarwal right in the beginning said, the aim or of this particular session is to guide all of you to be able to publish in journals, your target aspirational journals, journals which have a very good standing in the community, which have high impact, right? So we will be talking more about peer reviewed literature. So when we talk about peer reviewed literature, this is scientific writing. This is a piece of science that has been reviewed and validated by experts in the field, right? So even a grant application is reviewed, right? But it is not publicly available. Then we also have some non-peer reviewed stuff. For example, a thesis. Now, thesis, many of you might say that it is also peer reviewed, right? By your thesis committee, by your, guide, the principal investigator. But it is not peer reviewed in the form of a formal journal. What the peer review process that a formal journal article undergoes, that kind of peer review process is not. Then there are also letters to editors and sometimes some kinds of research reports which do not go through a formal peer review process. Many times, book chapters, especially those which are solicited, experts in a field are solicited to write a book chapter in a book. These are typically not peer reviewed, because the idea here is that he or she is an expert in the field, and therefore this is an expert contribution. It does need not undergo peer review process. So now, the first thing, we'll let us try to understand what are the characteristics of academic writing. We will be going into academic writing now in detail in the coming slides, but let us first talk about what are the characteristics of academic writing. It should reach the audience with the same meaning it has. Very important, especially a lot of you are clinicians. There should not be any ambiguity, okay, in what you are trying to convey to your audience. It should not be interpreted in more than one way. Remember, when you read a novel, uh, the hallmark of good literature, so I'm talking about non-scientific literature in the traditional sense, English writing, a novel, etc. The beauty of a poem or a novel is the different ways people interpret it, the different perspectives, right? That's why even today Shakespeare is so relevant, because people interpret it in different ways. This is exactly the opposite of what scientific writing strives to do. There should not be any subtext. There should not be any two different interpretations of what you're saying. There should be only one meaning. That is simple. Now, a lot of us, because we are Indians, we are not native English speakers, we are often a bit hesitant about writing, right? If you consider that scientific writing works best when you write simply, it actually will make our writing much easier. Just write small, simple statements that are representation of the fact. That's all. You should be concise. In fact, the Journal of Bacteriology, in the instructions for authors, it used to have a line which I used to really love. They used to say the best English is that which gives the sense in the fewest short words. Simple English, no, elaborate, artificial, ornate, or complicated words. But when you start with a concept, okay, this is a characteristic of academy. But where do I start with? You first start with a thorough literature search. Okay, why do I start with a thorough literature search? A good literature review demonstrates that you know the field. Okay. Now, over here, let me stop for a moment. Let us get some examples for from some of you. Can I have some of you unmute yourself and maybe come up with an example that you are of a project that you all are working on, and then we will try to understand why literature is very important.
Hello, ma'am. Good
Yes,
evening.
Yeah,
Uh,
hello, ma'am.
Dr. Anita here from Ramaya Dental College, Bangalore.
Hello.
Yes, ma'am. Can you hear me, ma'am?
Yes, I can hear you, ma'am.
Yeah. Right now, I'm trying to write a systematized review.
Okay, systematic review. Yes.
Yeah. Yeah, it is not systematic review. It's a systematized review because
Okay.
ये concept थोड़ा सा नया है. I'm
Okay.
just introduced to the implementation research concept.
Okay.
I'm writing an overview come systematized paper about process models in implementation research.
Okay.
Yeah. So
So I think can so now you said you are basically trying to write a review of all the process implementation models in research, right?
yes,
So for in your case, ma'am, I would say a good literature review is everything because you are not really presenting any primary data, right?
yes, yes,
Yes.
yes.
So in your case, there is a very important role of this. So here, Dr. Anita actually is a very unique case. She is working on what is basically she can call it a systematized review, a systematic, basically a review. Okay, she's trying to understand what has already happened in the field and come up with either a synthesis of the literature or some perspective, some kind of conclusion based on what is done. In her case, there is no primary research which is happening. In her case, a good literature review is absolutely important because until and unless she has reviewed the major studies or the findings in the field, how will she come to the right conclusion? Correct. Now, let me contrast a little bit with somebody who is working in laboratory based research. Maybe you are working on a new therapeutic regimen that you have seen working better in some patients. Why have you even come up with that therapeutic regimen, or why are you using, or maybe you are working on more basic research, okay, maybe a causal agent of a particular disease, let's say a particular gene. Why are you thinking that any kind of modulation of this gene will be able to cure the disease, or there is some kind of causal effect, or maybe this gene is a good target for a good therapeutic target for this particular condition. This is based on previous findings. In fact, if you go to any kind of and this is, I'm talking mostly from the biomedical field, you go to the NIH website, National Institutes of Health, United States. One of the things that they say, one of the reasons that many grant applications are rejected is because people have not done a good literature review. They don't know what has already happened in the field, and therefore they cannot come up with a good hypothesis. So, all of you who are coming up with a good research question, the major thing is a good literature review is absolutely important for formulating a good research question. It justifies the reason for your research in terms of publication, in terms of being able to publish in high impact journals. When you go do a little, see, your papers are going to be, your manuscript is going to be reviewed by experts in the field. The editors typically are senior people who have years of experience. When they read your the background section, which typically introduces the reader to why you have done this project, why have you worked on this particular hypothesis, how you have come to this hypothesis, this is where this is where the editor, when he sees your how you have come to that conclusion, that why this particular question I should be working on, they look at the references and they know what has been what has happened in the field. And if they don't see those references which represent the state of art in the field, that is one of the biggest reasons for desk rejection, remember. So one tip, a pro tip from us: make sure that you are quoting the most well cited, relevant literature in your field, in your paper, in order to make a positive impression on the editor. because that tells the editor that you know the field well, and also tells them that you have understood the field well enough to justify the reason for your research, and allows you to establish the theoretical framework for your study. uh a literature review we have put. Now these are some things. Let me try to, just give me a moment. As I go on, I am going to be copying these and I'm going to be putting this in the chat window. So just bear with me a moment. Um, we have a chat window. Yes, we do. Okay. So can you see I'm going to be putting in a lot of links, etc., in the chat window. That should be, you can just take, copy and paste them in a text document. These are good links to read on why and how a literature review must be done. So give me a moment once again to share. I think my screen is still being shared, right? Yes. So when you do a literature review process, clearly define which topic or field is being examined, and you find the materials. Now, in this case, what I'm going to do is I'll skip some of the next slides and I actually go into a demonstration of how you should be doing a literature review. Okay, so just give me a moment as I pull up a database. So I'm going to be using for today's purpose. The database that I'm going to be using is called Web of Science. Many of you probably have access to this database in your institutions. So just just make sure you talk to your library and see whether you have access or not to this. If you are a centrally funded research institution, most probably you have access to this database. Just give me a moment. I'll share it in a bit. Now, the reason why I am using a database like Web of Science, by the way, also hosts Medline. Now, the reason for this is very simple. This is a structured database, and this will help me retrieve articles much more easily than if I do a Google Scholar search. So just let me stop presenting. And let me Okay. I hope now you can see my screen, right? This is how your Web of Science, platform looks like. Now, I'm going to be using a lot of, operators, etc. So we are going to be doing a search, let's say, on renal cancer. Now, tip number one, when you're doing a search on a particular disease or any field, try to have as many different synonyms as possible. So if I have renal cancer, I should think about all the different ways renal cancer can be written in a manuscript. So if I take, for example, let's say I take breast cancer, so in that case I should say breast, or maybe mammary, because mammary tumors, and because I want them to be very near each other, so near tree, tumor, or tumor. And this is a common syntax. You can use this syntax even on PubMed. One small tip, please never do a literature search just on the internet. You do not know the quality of literature. There are several different publications which are low quality, what we call predatory journals. Always using a database that has some level of curation in order to do your literature search is extremely well advised. So let's say cancer or carcinoma or neoplasm. or hyperplasia. You can go on and on. So I'm going to get a huge number of papers, obviously. 679,000, right? Now, let's say the only thing interested in this particular, see, if you notice over here in these papers, I can quickly, now let's say I want to know about BARCA1, BRCA1 mutations. Okay. Now, out of those, I just want to know about genome-wide studies. So I can do a GWAS, genome-wide. Can you see? 110 only. And I can also put them according to highest in citations. So this is the what a citation database allows you to do. Quickly figure out which papers are extremely well cited in the field. So if you're writing a paper on genome wide associated studies of BRCA1 in breast cancer, this is probably a very important study that you should now, if you want to know about the new things, this is a 2017 paper, Association 65 New Loci. Now, I want to know about what are the new papers, related. Can you see this paper has been cited by 620 people? This is how you do a literature review. Okay, so but this is an older paper, 6 years old, right? They have found 65. Give it a moment to stabilize. Okay, so it has. And not only that, if you look at a database like Web of Science, they also tell you this paper is cited in the discussion of 51. These are probably the papers where they are using this data to inform the results in their paper. So not only now this public particular publication was cited by how many other different publications, but in which section of that publication this parent publication was present, right, as a reference. So see over here, now you have 51 references. You can quickly now find out, okay, 2023 paper on some microRNAs. Okay, this sounds interesting. So now I can find out which are some of the newest research which talks about finding new genes which put a patient at risk of breast cancer. Now, this is a kind of analysis which would have been very difficult if I had gone to Google Scholar. You can look back and forth in citations when you have a database like Web of Science. Okay, so I am not saying that Web of Science, of course, because I am from Clarivate and Web of Science is a Clarivate product. I am using this as a guiding principle for you to show. But my um, urge or my persuasion is over here to all of you. My advice to all of you is use a structured database. Also, when you use a database like Web of Science, you can always create lists and libraries and folders. You can arrange your results in a certain way. Now, let us say you do not have, just give me a moment. Let me come up with a, okay. You can do the same kind of search on the Medline section. We also have Medline on Web of Science. So if you are working on Medline, right, you have a mesh heading, right, and a mesh major topics. So you can go to mesh major topics and search for breast cancer. This is a great way to again find different synonyms. Find, okay. Can you see? I want to search only for the BRCA protein. Details, BRCA protein, BRCA1, add. Search. 7,000 results for BRCA1. You can also search the publication type. So whenever you are working on a literature review, you can always, so you want to know something about a guideline, there's a one on guideline. Okay, recommendations for the medical management of, okay, this is an old paper. You can always, again, this allows if you are on a system like Web of Science, can you see you can, so why I am telling you to use a database like Web of Science, there are other databases also, but the reason why I'm asking you all to do your literature review on a structured database is one, allows a lot of analytics and filters so you can actually go get to your relevant data set. And secondly, another thing that you can do is you can export them onto any kind of a you know, so you have EndNote Online, which is a reference manager. There are different reference management systems. There are systems like Mendeley, Zotero. Remember, if you are a researcher who is using Web of Science for their literature review, you can always export these references to any other reference software using this particular filter. So this filter works with BibTeX and RIS and other things. Okay, you have more export options also. Okay, so with this, I'm going to stop presenting and I'm going to come back to my slides. But I hope this gives you an idea on how to do a literature review. Okay, be always do your literature review in a top down approach. Yeah, Dr. Anita, you have a question?
Ma'am,
Yes.
that's clear enough. Thank you. I just have a small query here.
Yes.
Now, you can export that as RIS, you
Yes.
said.
Yes.
Just demonstrate that part once, ma'am.
Okay. So I don't have anything other than, just give me a moment. I have another question from Sunita Kalta. Sunita?
Yes, ma'am. My question is not regarding the current slide. Actually, it's on the second slide.
Okay.
Regarding, you have said that it should be, the interpretation should be a single way.
Yes.
It
Yes.
should not be have many kind of
Yes.
interpretation. So, ma'am, can you give one example for my understanding?
Okay, so when you say things like, do not say we in the conclusion, okay, when you're writing the discussion of a paper, don't say things like we hope or we believe that these results will be instrumental in, you know, or use words like paradigm shifter. How many times have you come across these kind of statements in papers? We believe paradigm shift. These are words and statements which are very, how do I put it? They are very weak, right?
Yes,
I
ma'am.
I have so we have a magazine called Bio World, and I contribute to it. So my editor, who is also a PhD, she's a scientist, she is also a biomedical scientist, and also she has taken a science communication degree from Harvard. She once told me, until and unless you are a psychoanalyst or you are somebody who can see into the future, clairvoyant, do not use words like the authors believe and all. You don't know what anybody believes. Nobody understands what you believe, what you
Yes.
think. Okay, so my point here is always use terms which are concrete. Don't say that we might, don't use might. This can have two meanings, right? Okay.
Yes.
Secondly, in the discussion, and this is something I'll come when we discuss the best practice of writing, don't be overtly enthusiastic. So many times, we write papers where we've seen some results in a small population, small cohort, or we have seen something in the laboratory in animals. You cannot say this is a new cure for a particular disease, right?
Yes, ma'am.
And this is also bad science. I'll give you an example. How many of us know about ivermectin, right? In COVID, a lot
Yes.
of ivermectin clinical studies were done on the fact that ivermectin was shown to kill certain increase the virus load in just petri dishes. No preclinical studies were also there. Just based on those results of killing cells in a petri dish, people extrapolated it to human. And there were doctors who actually prescribed ivermectin. Before World WHO, CDC all came up with advisory that there is absolutely no clinical proof, okay, that these drugs are able to, they are virucidal, that they can kill the coronavirus, right?
Yes,
So
ma'am.
the point is, many times when we write this, we are sending a wrong message to the community. We are over exaggerating, extrapolating, and giving different meanings. So when you write a paper, the meaning should be concrete.
Okay.
No thinking, nothing. Our results show that this works in humans. That's why many good journals today actually have a section called limitations of the study. How many of you
Mm.
have seen this in journals, limitations of the study, where the authors have to tell that these results are in a small group. These results have been only in this kind gender, or these results are in, they have not seen these results in any other model. They have seen it only in a particular one animal model, right? So
Yes, ma'am.
you have to, this is good science when you tell people, when you give everybody an absolutely correct picture of what the results mean. Does that make sense?
Yes, ma'am. Ma'am, also during discussion, we
Yes.
sometime in our result,
Mm-hmm.
we try to, when we interpret, we give different author citation, like this
Yes.
paper is
Yes.
saying this
Yeah.
and this paper is saying this. So there also sometimes we are giving different kind of interpretation. So in that scenario,
See, that time you're not giving interpretations. You're not giving it.
okay.
Because these are already what is there in literature, right?
Yes, ma'am.
This is not an interpretation. This is what the
Hmm.
author has already said. You
Okay.
are interpreting your results in the light of older research, right?
Okay. Okay. Okay.
Okay, so
Get
that
your point,
is
ma'am.
something I'll discuss. So just to
Okay.
come back
Thank you.
to Dr. Anahita's question,
Thank you
uh,
so much.
ma'am, this is how you'll export it. You will get an export. So I am going to select, let's say, author, title, source. Okay, and export it. I'll get a file over here. Okay, and this file you can open it in Mendeley. Can you see, ma'am? .ris.
Yes, ma'am, I'm seeing.
Okay, so this is how the file will be saved. Now, in case you're working on Google Scholar, right? Now, this is another thing that I wanted to show all of you. If you are working on Google Scholar doing some searches, remember you can always create your library. How many of us create a library on Google Scholar? Can I have a show of hands? Anybody who creates a library on Google Scholar? Nobody?
No, ma'am.
Okay.
We all
Okay.
just directly based on the requirement of
No,
our.
so you can always create a library, and creating a library is a great thing because that can also help you import to different formats. So let's do a search on, let's say, urothelial cancer, carcinoma of the bladder, and I want some papers. Can you see this over here? You can save everything, whatever you want to save a paper, you can save it, and you can even give it a label. Okay, so you can create a label. I can say this is called urothelial. I'll tell you why this is a great feature which has been introduced in Google Scholar. Let me save two papers over here. Done. Now, can you go over here to your library, and in your thing, urothelial cancer, these two papers will come, right? And you can export all of this in EndNote. You can export in CSV, BibTeX. I think BibTeX might work with Mendeley, but on Google Scholar you have an EndNote option. And by the way, EndNote, all of you who have Web of Science, you know that the online version of EndNote is freely available to you all. This is how EndNote looks like. Let's give it a moment.
BibTech works with mainly Google Doc, ma'am.
Okay. So I'm not sure what works with Mendeley, but this works. EndNote, Google Scholar has a EndNote import file, so you can always do collect and import references on in EndNote from different places. So if you all have Science Direct at your institution, Then Science Direct, when you are looking at papers, they allow you to, import citation. If you are importing from Science Direct, use refmin-ris. If you're importing from Google Scholar, use EndNote import. Go to your downloads. Can you see? Saved Rex one. No, I think this is in the RIS format, right? So let us do refmin-ris. Select. You're going to select anything. Book chapter import. So hopefully this will import from the refmin-ris. So going forward. So to answer the question to Dr. Anahita, does that work? It'll take some time probably. The correct format typically is EndNote. So export all in EndNote. EndNote import. Choose file. Download citations. I don't- It's taking some time. We can come back to it in a moment. So stop sharing. Okay. So let's go. These are some of the different. I have already talked about. It should be an iterative example. Can you see different ways we have? Once you identify, come up with it. This is something you can take a screenshot. This is some of the rules of how to do a search. When you are trying to decide which papers to cite, some of the questions for critically appraising is with what question is addressed, what are the key concepts, what are the strengths and weaknesses. So if you are citing a paper in your research, okay, as ma'am, Sunita just talked about, interpreting other researchers' work. If you have two researchers who have worked on the same topic, but you see and one researcher having results and data from human studies that is a higher level of evidence, right? So when you are looking at the research that to cite, see ones which have a higher level of evidence. Just like in systematic reviews and meta-analysis, you have the higher level of evidence, when you choose case reports versus cohort studies, etc., right? So the level of evidence you have to critically appraise which paper is giving me evidence that I can have more confidence in. That is the paper that I should be citing for a particular statement of fact. Makes sense to everybody? Now, talking about the major techniques in scientific writing, we have something which is a definition. Okay, so here I am looking for original research papers. I am now not going into systematic reviews. We at Clarivate actually did a very detailed webinar on systematic reviews in evidence-based medicine. If any one of you wants a recording of that webinar, please let me know, write to me, and I will send you that webinar, on-demand webinar. Okay, so major techniques in scientific writing. A definition, one is definition. When you're defining a disease or some new disease which you are describing, okay, it is more a descriptive way. Typically a lot of review articles are like that. However, it can also be the first report of a certain, let's say, a case report. Okay, that could be also written in a definition where you're just describing what are the symptoms, what are the different aspects of the case that you were, you're reporting, of what are the presentations, the symptoms that the patient had, how did you treat the patient, etc., right? You are not, there is no comparison with previous literature in that, uh, you know that kind of writing. Another kind of writing which is very common in application based translational sciences, you have come up with a new drug. you are comparing it, contrasting it with. So let's say the standard regimen in breast cancer typically has been doxorubicin along with some kind of a HER2 inhibitor. You are now putting your patient onto a new, maybe some kind of immunotherapy along with doxorubicin, some new regimen. So comparing and contrasting, right? So you will have, when you write, you will say, why have you come up with this regimen? Because you will now identify gaps in literature. Does everybody understand this concept of compare and contrast? Enumeration and illustration is very similar to definition, and so is cause and effect is when you have a certain cause, and for a especially in biomedical sciences, you have a particular, let's say, gene. You are trying to say that this causes, let's say, mutation in the gene causes this particular disorder. You're describing what is it, basic science. You're not comparing and contrasting. Many papers are cause effect plus compare and contrast. Okay, so maybe I'll give you an example. I recently reviewed, we were writing a piece on this paper where it talked about a designer bacteria. They had modified some apparatus in E. coli. Okay, and they so that E. coli can carry a therapeutic payload. It is carrying a TNF alpha antibody, and that TNF alpha antibody, once you are giving this E. coli orally, this designer E. coli to the patient orally, they will release the TNF alpha antibody only in the gut, and this can decrease inflammation in IBD, inflammatory bowel disease, in colitis. We all know when you give antibodies, especially TNF alpha cytokine antibodies, systemically, infliximab, etc., there are a lot of different side effects, adverse effects are there. Immunocompromisation takes place. This is supposed to reduce the side effects. Now, a major part of this study, when they were so this study, they are talking about how they have created that platform. In that case, that platform, when they're talking about, it is in the enumeration illustration way. They are talking about how they have created the platform, how have they engineered the E. coli, how they have introduced another, they have transfected a genetic apparatus into the E. coli that will allow it to take a payload, and then it can get inside the body and release the antibody at a specific site. That is like they are describing the system. But the second part is about compare and contrast when they're using it in a animal model of colitis, and they are showing that how, when you give this particular drug delivery system to this animal model, it is better than systemic administration of TNF alpha. Can you see, a paper can have multiple, right techniques. One part, it can be used for, enumerating and illustrating a new concept, and then using that concept in the light of older research to show that it is covering certain gaps in research. Does that make sense? I'll just pause for a minute that you have understood this concept. Any questions? Okay. Now, before you start with a manuscript writing, there are 10 key questions that you must be able to answer. First question is there a story to tell? What was the rationale for the project? So the first thing, I'm just going to use my,
Presentation mode, ma'am.
yeah. So when we have these things, is there a story to tell? What was the rationale of the project? This particular place, this is covered by your, this minute, sorry about it. First, so you should actually have this checklist on your, hand. Take a printout of this checklist. Is there a story to tell? This is when you have enough. Whenever you are doing experiments, first thing you should do, a pro tip is, first thing that you should be doing is, first make a point of ensuring that they tell a story, so that you have a to B to C. There is some story is there. Okay. With whatever results you have in your hand, okay, try to see whether I can flesh out a manuscript or not. Many times when you start getting rejections from journals, that is when you realize that I did not even have a story. I did not have enough data to tell a whole story. So first thing, once you have got the results, you have been doing some experiments, first thing should be trying to put the results in a logical order that they tell a they try to explain some phenomena, they try to tell a story. That is the number one, because then only you will come to understand that are some experimentation missing, is some critical piece of analysis or data missing. Then you come and then this is where you should, be able to, because this is also for a grant application or any project. Why was I doing the project? Now typically this is something that you should start, even when you before you invest effort and time, you should have this in your hand. So I do not know how you all are doing your, like when you have how do you do your experimental design. The best way to do an experimental design is actually have a good idea of how am I going to proceed with. I have a broad area that I'm thinking about. Okay, maybe my group works on a particular disease, it works on a particular genetic component. I know this is the area I'm going to work, but what area am I going to work? You first do an exploratory literature search. Okay, try to find out the gaps and then maybe pick up an area, and you discuss with your PI, with your other seniors, peers, that this is the area of research I am going to work on. And then you and then you decide, why should I be working on this project? Because there is a particular demand, this is a question that has not been solved. You should have a rationale. So typically, I mean, the best practices have the rationale even before you come up with anything else. Did I perform a thorough literature review? This is something that you have to be very careful. If you are doing literature reviews on databases like Web of Science or any citation database, they allow you to save your queries. So if when you are trying to write your paper, maybe you did the literature review in order to identify gaps and all 6 months ago, a year ago, it is a good idea to again redo the literature at uh, you know regular intervals of time to be updated, to be abreast of the new updates in the field. Did I have a clear hypothesis? What do you mean by a hypothesis? It's nothing but the research question. Do you have a research question? You should be able to write a one-line research question. For example, a good research so if I have a research question, I say what are some of the effective strategies for uh, preventing smoking or for cessation of smoking in young individuals? Is this a good question? Can anybody tell me? This is a good research question. Anybody unmute? This is
Ma'am,
yes.
please, sorry. Your
The
voice is breaking. Please repeat,
yeah.
ma'am.
The question that I have come up with is, what are some of the effective management strategies to stop smoking or for cessation of smoking in young individuals? Now, I want to know, is this a good research question? It is not a good research question. I'll tell you why.
It is not. It is not.
No, ma'am, it is not a good research question.
Question, because it is not a definite research question.
It does not have a direction also.
Yeah, it doesn't have a direction. It is not a definite question. You cannot
Also.
a hypothesis. I think somebody should unmute.
Everyone, please mute yourself and raise your hand. We will call one by one otherwise it will echo. So Google Chrome, Google Meet has this limitation.
Yes. So a clear hypothesis is one where the question that you are asking can also be falsified. So today's session, I cannot get into null hypothesis and all of that. I'm sure many of you know about it, but basically the hypothesis is something is a question that I can falsify, right? I meaning that I can have one answer that is that no. So if I say um, social media awareness, okay, or like awareness, so social media, this is a effective management strategy for stopping smoking in young people. That is a good question, because now I have an alternative that no, and I can run a statistical test by saying yes, this is a good I can present data, experimental data, that this is a good strategy, or no, that this is not a good strategy. So I can have two groups. I can have a group that does not look at social media, that does not, that was not shown any kind of, they were just given they just got the information through statutory warnings on cigarette packets. So they you have one group that does not have social media exposure. They did not come across campaigns, etc. Then you have one group which was using a lot of targeted social media sites, and in those sites maybe we did a lot of advertisements and public service announcements about the disadvantages and the uh, cons of smoking, and how the two populations now behave, right? So that is what is a clear hypothesis or a good research question. Then you design the experimental design, the survey, how will you collect the primary data? Is the experimental design defendable? Have I the right statistical tests? How am I going to treat the data? How will I justify? How will I use? So please have this checklist. Every time when you start with a manuscript, before you submit it, even to your boss, make sure that you are able to give a yes check mark to each of these questions. Writing the paper, key points. Strong introduction, yes. Always start with a strong introduction that engages the reader, sets the scene. Clear, logically organized, complete methods, results, and relevant discussion. And now with this, I am going to I'm going to let you just read this, what editors look for in a manuscript. But now I am going to go one by one. So I just let you read these slides. But now the major point I am going to do is go one by one, how to write an introduction, how to do the methods, results, abstract, title, discussion, etc. So first thing about the writing style, reduce dead words. So do not use words like very. Do not use words like really, basically, generally. We do use it. We do say typically, but when you're writing, if you have these kind of adverbial phrases, take them out. As I said, short sentences. Always use subject, verb, object, meaning active voice. Passive voice is to be avoided in most sections of the manuscript. And use strong verbs and avoid turning verbs so into the nouns. Eliminate negatives. So we did not want to do this. Try to say it in a different way. Use positive constructions. So this is the article structure which we call the IMRAT format. Okay, introduction, methods, results, and discussion and conclusions. Now, typically, when you start writing a paper, always start writing with the methods first, because that gives you an idea. If you remember the checklist, okay, the last 5 questions about the proper appropriate controls, experimental design, they all relate to your methodology. So if you write the methods first, this is where you'll be able to understand whether you're hitting those points in the checklist or not. Once you have the methods, then you start with the results. Remember the first thing I told you, if there is there a story to tell, that's when you all the results that you have accumulated from your, studies, you Arrange those results so that you can tell a coherent, logical story. Once you have the results, right, then you start writing the discussion and the conclusion of the study. Introduction comes last. Never start with introduction. Okay, you should have in your mind why I did the study, but sometimes you can write a better introduction and justify the rationale of your project if you have written your discussion and conclusion first. Introduction or background. Now this is a critical showcase. If you remember in our pro tips, we told you that you must be able to write a strong introduction that engages the reader. Okay, and what should you not do in an introduction? Do not overtly state the research area. So if you're talking about, like, let's say it is about, disease, even COVID, don't try to write everything about COVID. Now people who are who are your audience, remember, you do not treat your audience as if they do not know anything about the subject. They are also, so this is not a layman. Scientific writing is not for the layman. This is not being read by somebody who doesn't have a background in that subject. This is going to be read by other scientists, clinicians, practitioners who also work in the same field. So they have an idea of the research area. So do not overtly state and include textbook information or overt citation. Description of results and methods from published studies or present studies is important. So if you have, what are the right ways to cite? If you have older research, yes, cite them. But if you have a newer review that encompasses all the older studies, put that. Do not do citation dumping. Citation dumping is when you have one statement and you have 30 people just put 10, 11 references for one statement. If I say MDM2 is a regulator of P53 and a critical oncogene, and I start citing references from 1992 to 2020. So what am I trying to tell my reader? What is important? Either I just say take the original research article which proved MDM2 to be a cancer-causing oncogene, or I cite a recent, well, a review in a high impact journal for that. Inclusion of references that are not generally available. Please avoid obscure references. Another thing, do not be overtly negative to studies of other groups or overemphasize studies only from your own group. Introduction is not the place to introduce any kind of figures or complex tables. No, you can probably have a sort of a graphical representation of the problem, something like that, but typically no complex figures or tables. Materials or methods. This is usually specific to the journal. This is where you can write it in the past tense and the passive voice because you've already done it. Now, methods and materials is place where you can end up with a lot of self-plagiarism, because let us be fair. Not only, you might think that I am citing from or I'm writing in the style of my own paper, but self-plagiarism is also a thing. And how many ways can you write a particular method of protein estimation or a certain ELISA, right? So in case you are taking a method as is from a previous publication, or it is a well-known method in the field, just say this method is taken from XYZ 2015, whatever that, just put the reference. There's no need to describe the method. It is okay. If you think that you have some methods which are extremely complex, and you have, like, it could be a gene transfection method, etc., then in that case, or maybe a surgical method which you have to describe in detail, that is something that you need to put in the supplementary, because many journals have a page limit, a word limit. So much of the section can actually be included in supplementary data, because if somebody really wants to replicate your method, they now know where to go. Okay, and some journals, they actually have the methods after the discussion section, example Nature and Science. But please describe the statistical study design and procedures for analysis, and this is very important, because this can be Remember, your methods and methods like a recipe. It should allow other people to replicate. This is the whole point of science. It should allow other people to replicate, understand the context in which you have got these results. Let's go to results and discussion now. Present selected data, giving the big picture. Very important. You have done 20 different experiments, you have a lot of results. Everything does not need to be given in the result section. So you might have come to one conclusion. Many journals ask you for several confirmatory experiments, etc., to give you the same, you know, question. Sorry, to confirm the same, point, right? So you might have got one, you have done a more simple assay, then you have some, very sophisticated confocal microscopy, electron microscopy, etc., some very genomics experiment to prove the same thing. You decide which data you should be putting. No need to put all the again, lot of results can go to supplementary section. You should present the data that gives big picture, okay, and which can be interpreted. If you have certain results which are not, until and unless they take away from the interpretation, okay, if it is not really making, it doesn't make sense to put it, don't put it. Refer to the key published data. And again, remember, results is function is to relate the present work to previous reports and point to future efforts. Okay, do not repeat your introduction. You have also in the result, do not start with the same thing why you did the study. You have already talked about that in the introduction. And remember, a good results discussion is if you have presented result 1, 2, 3, 4, do the same way in the discussion 1, 2, 3, 4. Explain the significance of the results uh, in the same order as presented in the results. Another thing, the length of the discussion is generally proportional to the word of amount of new information. So 1/4 of the manuscript. And do not make grand statements. Okay. So for example, this novel treatment will massively reduce. No introducing of new terms. So if you are introducing any kind of terms earlier in the paper, so defining the term for, let's say, TNF alpha, you have to say TNF is tumor necrosis factor right in the beginning. Okay, in the introduction, not in the discussion. Discussion is not where you introduce new terms or give the full form of the abbreviation, etc. Do not use unspecific. So if you remember when I talked about different meanings, etc., unspecific terms not to be used. Higher temperature, lower temperature, no. Use quantitative descriptions. This is a great blog for all of you. Just a minute. I just want to give you I just want to put this in the chat. This is a wonderful, it's an old writing, 1990, but it is something which is, I mean, it has been used by scientists all across the world for a very, very long time. So, just a minute. Somebody has asked if you is it okay if you use PubMed and Mendeley? Yes, absolutely. Mendeley is a reference management, madam. It is not this thing. A format for Mendeley would be RIS-RefMen. Okay, and Mendeley is also a good reference manager, so you can use that. That's not a problem. Is paraphrasing allowed using paraphrasing software? Okay. Remember one thing. Few days ago, if you read a blog called Retraction Watch, a paper was retracted because they had extensively paraphrased another paper. So do not paraphrase entire sections of papers. That's why I strongly recommend not using AI tools, for these kind of purposes. because these are again, just like you have paraphrasing software, there are softwares that can actually de-paraphrase and understand what is the source title. Okay, so do not take entire sections of other people's paper, just run it through a software and then put it. These can be caught. Just give me a moment. So again, these are some of the do's and the don'ts. Use figures and tables to summarize the data, show the results, confirm the method is reliable, tell them why you have chosen the method, and define the limitations of methods. Do not duplicate data among the tables, figures, and text. That becomes data fabrication. Please remember, if you have a Western plot, you cannot use the background in for two different. Okay, so be very careful. Use graphics to illustrate data that can be easily summarized with text. Figures and tables are the most effective way to present results, but remember, your legend should be able to. So I remember my PhD boss telling me, you should be able to understand a paper by just looking at the figures and the legends. That's it. You should a well-written paper. There is no need even to read results and discussion anything. You should be just able to read all the panels of figures and understand what the scientist is trying to tell. So make sure that is how your that's why legend should be standalone. That there is no you the no re, the reader should not have a requirement to read the entire manuscript. Another thing, this is about economy and money. Lot of journals actually charge you if you have to use a color printout. They have to put color. So so if you're doing some PowerPoint figures or any figures that you're drawing, make sure you use colors which are have enough contrast between them so that they can be rendered well in black and white also. If necessary, of course, color have to be used. For example, if you're doing a fluorescence imaging kind of a thing, in Illumina, the IVIS, where you actually show the heat map, there you have to use a color. But otherwise, if color can be avoided, make sure you use colors that come or are depicted well in black and white also. Abstract, indicative abstracts. You can also have structured abstracts in medical journals. They actually tell you, I've seen in a lot of medical journals, the abstract also has introduction methods, so that is in medical journals. This is the purview of the journal, absolutely. But only thing, the tip is, abstract is the one which is used to promote the article. Remember, when I look up, do a search on Google Scholar, I do a search on PubMed, I do a search on Scopus, Web of Science, the presence of certain buzzwords and keywords, that is what is in the abstract. Make sure the abstract comes in my search, right? The paper, the manuscript is retrieved in my search. So good abstract should be standing alone, brief and specific. Do not use technical jargon, but use buzzwords that will pull your abstract in a search. Good title should contain fewest possible. Do not have long titles. Have nice, catchy titles that describe the contents of the manuscript. Do not use unnecessary jargon. Do not use uncommon abbreviations. Do not use unnecessary detail. For example, I talked about that, E. coli. A good one would be designer E. coli reduces inflammation in gut. Okay, the designer E. coli can deliver the TNF antibody to reduce inflammation in gut. That's it. Tells you everything that you need to know. What is not needed is, don't say preclinical model of IBD. You can just say reduces inflammation in gut in mouse model. That's it. With a moment, or you can say reduces inflammation in mice gut. That is enough. The moment somebody reads mice gut, they know you are talking about a preclinical model. So can you see how I'm having a shorter title, which is still complete? Keywords, check the guide for authors. Very important. Again, for every journal, please check the guide and the scope. That is going to be very important. Good citation practices. These are some of the general suggestions. Limit the number of citations to fewer than, if it's an original research article and you have to cite 100 people, that means there's some that your research is not that original. Okay. For references, if possible, use primary resources, especially if you're talking about data in terms of numbers, like epidemiological data. Okay, especially in public health, etc. If you're talking about epidemiological data, etc., use primary sources if possible. Okay, I have already talked site reviews. You can always have as summarized as reviewed by in methods and materials. You can, if you are taking a method as it is from another paper, just write as this method is taken from so and so. If you are modifying the method, you can also say this method is being modified from Smith et al 2050, and then just put the modification part. I don't have to give you the importance. Please avoid excessive cell site. That can be one red flag which prevents your paper from being accepted in a journal. Check references for accuracy. Do not put references to unpublished data. Very important. You can put unpublished data in the body, but in your bibliography, there should not be any mention to unpublished data. Do not cite more than 3 references. I've already talked about this citation dumping. How to choose the right journal? Decide where to publish. This is something very important. Do not look at these kind of, if you see all these global impact factor, universal impact, and especially some journal, if we just use the pen. Please remember Index Copernicus or saying that this journal is there on Google Scholar. These are hallmarks of what we call predatory journals. Okay, so do not use them. These are some of the practical guidelines towards identifying predatory journals. Um, Another small word of caution, this is especially for biomedical scientists. Many open access publishers today are using the guest editor model to come out with a huge number of publications. One example is MDPI. Recently, one of their journals was delisted, by Web of Science. The guest editor model lends itself to a huge amount of malpractices because there the core editorial team does not have control over the peer review, and there can be, and especially many of these open access journals have very short turnaround time, so be very careful about these journals. Having a decent turnaround time of at least a month and a half, make sure that the peer review is done is rigorous, and that helps in improving your content. Another place where you can actually look for a lot of good articles, full text articles, are places like the preprint repositories, medRxiv, bioRxiv. So that gives you a lot of information about that. So if you're doing a literature search, though everywhere you cannot cite them, okay, you should not be citing preprints, by the way. Many journals will not allow you to cite something which is not yet peer reviewed, but it gives you an idea about the state of recent research. So with this, I will finish up my session. Okay, and I think we have a good 10 minutes to do a discussion. Okay, I know this session has not been probably able to explain everything. What about curious? Is curious also predatory? Okay, so curious, as of now, I know it is indexed in Web of Science, but as I said, there are certain small things that you should look at. Any journal that publishes too many issues a year, that is publishing through the guest editor model, and that is publishing on too many different topics in the same journal, be careful about them. I'm not saying that they are predatory. These are journals where there is a very huge chance that the quality of peer review may be compromised, if not now, in the future. Okay, because it is logistically not possible when you have these models. These are not sustainable models in terms of good, rigorous peer review. So I'll now be able to take questions. Dr. Padi, any questions?
No, ma'am, going well, fine. You have discussed so many things in this short period of time. We understand it is a very complex subject, but you have touched many points, starting from citation, literature source to what to write, what not to write in the manuscript. Now it's open for the participants. If you have some specific questions to this talk, please ask. Please raise your hand. I will ask one by one. Dr. Deepjot, you have raised your hand.
Yes, so, ma'am, I just have a question about discussion, how to write the discussion well or
Okay,
crisp.
okay, yeah. So I think I will just reiterate some of the points that I made before. Okay, so some of the so I'll go point by one point for the discussion. Okay, the discussion we realize is a section where the authors explain the meanings and implication of the results. Okay, so results, you have already put your results. Now you have to explain the meanings and the implication. So and you have to tell the importance and the value of the work. No, now the first thing what you should be doing is, in a good discussion, you should not be repeating what has already been said in the review of the literature. No need to review, do that. Relate the questions to the questions that were set out. So in your introduction, you said what you are going to do, right? Why have you started with the project? What are you going to do? So you now your discussion should try to answer all the questions that you were trying to answer when you wrote the introduction. That what are the questions that we are trying to write? Show how the results and interpretations agree or do not agree with current knowledge. You know what, this one, just give me a moment. I will actually like to take up a very nice paper that we and use it as a reference to show how a good discussion should be written. Just give me a moment. Let me pull up that paper. And okay, just give me a moment. Let me just share. Okay. Um, where do I share now? Yeah. Okay. So this is a paper, a new paper. It is on what we talked about, engineered Escherichia coli. Okay. So just see, very easily they have told you small short titles. Now let us look at their discussion. First, let us look at their introduction. Okay, this is a from a very good journal called Cell, Host and Micro. So now let us look at the introduction. Let us look right below. Can you see what they have said? Here we report this. Okay, and numerous gram negative. We and others have previously established are functional. Here we report something, and then they tell you. Okay, so they have told you what they have reported and what are these, and they have said these studies provide the foundation of a versatile therapeutic platform. Okay, now let us go back to the discussion of this paper and let us see how have they written the discussion. Here we describe the development, okay, using synthetic biology. Okay, further we have developed, so they tell you exactly what they have done. Okay, and now see how they are relating their work to other groups. Other groups have also engineered microbes. Okay, and they tell you what have they. However, this is a gap. Can you see how they have explained that this is a gap? They start by in their discussion saying, describing their results. Then they are trying to say what have other groups done, and then they're explaining the gaps, and then they are explaining what else. Now see, a good discussion. Protect TS could be endowed with a on switch. This is something that they have not done, but they say this can be done. Okay, why they are saying that this can be done? Because they have already demonstrated some versatility. So they are now giving a forward looking statement by But why are they giving this statement that this can be done? Because they have already done something. So they are saying, I have already done 1, 2, 3, 4. Based on these results, we propose that 5 can also be done. Then look at it. They are giving you more information why their study is a good study. In studies similar to ours, they also observe. So they are now defending in their study in the light of other studies. Okay, and then they are telling you why this study is important to science given its inherent implemented in grass. Okay, however easy and this that we are also doing this. So are you seeing what have they tried to do? They have tried to say what have they done, why have they what do their results mean, what are the far reaching implications of the results. They are defending their results based on the previous findings of what is already reported in literature, and based on what results they have got, they are proposing that this is something else that we can do. This is how we see our results, our findings revolutionizing or taking the science forward. Does that make sense to you, sir?
Yes, ma'am. Thank you.
Okay, so just to recapitulate once again, what does the discussion should be doing? As I said, not repeat what has already been repeated in the literature review. Relate the results of questions set out in the introduction. Indicate the significance of results, explain the theoretical background of the results, and suggest future research that is planned or needed to follow up. So, any other question?
Next, please, Piyush Agrawal.
Ma'am, I have a small doubt. Like many
Yeah.
of the clinical journals, what
Mm-hmm.
they have a high word limit, like 4,000 words. But
Yeah,
if
yeah.
we see the abstract, it has a, and especially if it's a structured abstract, the word limit will be 250.
Yes, that's
So
correct.
every section should be like, if there are 5 sections in the structured abstract, well, that should also be like 50 to 100 words each. So
I
how
would,
to?
yeah, I would say that yeah, 50 to 100, but in that also you will have an introduction. Introduction, keep it within 50, like 1 or 1 to 2 sentences. Per in the abstract, put little bit more about the discussion part. Okay, I
Okay,
mean,
but it
discussion
becomes a
in the
bit
sense,
difficult to.
yeah, I do
Uh,
understand. I do understand, but this is what it is. If you'll notice, the journals, they most people try to economize in the first parts. So they don't write why are they doing the project. Very shortly, they write that. Typically, they give you right in the introduction. They put more emphasis on the statistical tests that are used and the results that they have, results, the meaning of the results that they have got.
okay, ma'am. Thank you.
So
So
anyone wants to ask, please unmute yourself and ask.
So there is a question in the introduction part. What should I be doing in order to engage the reader? Introduction should lead logically to the hypothesis or principal theme of the paper. You should keep it. Do not repeat the abstract. Avoid extensive literature review, and do not repeat textbook information. Always advisable. I'm just going to put some points in the chat. This is to Sunita, who's asked the question. Just give me a moment as I type in. Best written in active voice. Okay, so Rita, I've written the answer. Hope that helps.
So Smita ma'am, you have some question?
I actually do not have a question. Thank you, Dr. Subhasree, for such a lucid presentation and so informative.
Thank you.
Uh, now I know where to send my manuscript for a friendly check. So which is done free of cost. Oh, wonderful, wonderful.
Thank
I was, I mean, it's really, really wonderful to listen to you, and And I actually have a issue to discuss. Basically, it's a open question to everyone also. Like, where, how to decide? I mean, you have money, you can publish in open access, and
That's a great question, ma'am. I'll just tell you something. Today, all over the world, especially in biomedical literature, there is a growing, trend that, for example, in the US today, any grant that has been given by the government, right? That means any NIH, any federally funded project, that has to be made publicly available to the taxpayer, right? So, to all the people who are attending today, ma'am, and even Dr. Patnaik, who's asked the question, please remember you can always upload your final accepted version of your manuscript onto your institution repository or to your personal homepage o- as soon as publication, or you can even put it into a repository like bioarchive, okay, saying that this is a postprint of my find. You just cannot put it in the journal form. But uh, databases like Web of Science and all, what they do is they collect this postprint. They collect the full text of your manuscript and they attach it to the record. So why I'm saying this is that even if you publish in a behind the paywall journal, there are avenues available to make your research freely available to the public. There is no need to publish in an open access journal per se, just to make your article more reachable, the outreach. Does that make sense, ma'am?
Uh,
yes. So you mean to say you may not necessarily wait for a tedious peer review process in a good
No, no,
journal?
no.
No.
I'm saying you have to wait peer review, because what I'm trying to say is that after peer review, you'll get the final accepted version of the manuscript, right, ma'am?
Mm-hmm.
You can upload the final accepted version of manuscript even if the journal is a closed journal, behind the paywall journal. So
Okay.
effectively, what you're doing is you're making your article freely available to everybody.
Okay. I think I can relate to this. Many a times you see on the internet that the same thing is available as a with the PDF and with the
Yes, and you can also buy it from a publisher website.
correct. Thank you
but the,
so much for giving us this solution, because not everyone like us who can pay for it, and
Absolutely.
many a times the funding agencies these days they are kind of keeping a provision of asking for funds, which is for publication of the results, but
Mhm.
they are very limited.
Yes, but there is this because this option is there of uploading to any repository and all, and there's a great website called Sherpa Romeo. You can check the copyright policies of any journal over there. So Sherpa Romeo, please go there, check the copyright policies. Most of the times you'll see that there is a free way to make your article open access, and they tell you what are the copyright policies. So please follow that. What I'm trying to say is that in order to publish, it is not necessary always to spend a lot of money.
Thank you so much, Dr. Shubhashree. It was great listening to you. Thank you.
I think we have a question from Dr. Jose.
Yeah, ma'am. My doubt is
Yes.
regarding our discussion part.
Uh-huh.
Yeah, now I felt like I should relate my to my objectives of the study. And second is what is the new which one I have done from others.
Yes.
Then the third part is what is the importance of the study which I have to in a current scenario. This is the third
Yes.
part.
Yes.
Fourth part, you told me what is in the future.
Yes. What you
I didn't I
plan?
didn't I didn't get in that in
Yeah.
exact
So,
what you mean.
so what you plan? So so I was giving the. Yeah, can you mute yourself, sir? Yeah, so I was giving the example of the paper that I showed you, which was on a designer bacterial system to deliver some drug. Now, they have done the study in a preclinical mouse model. So, the obvious future thing is that they have to do it in a human model, right? So, future means what? How are you going to take the results of the study of the what is published in this study further? Because no results in science can be viewed in isolation. They should form the backdrop of another study. So, how do you propose to take them up in the future? Are you doing some planning? Putting that in a paper gives your editors more confidence that you know what you're doing. You have a plan to take this. It's a continuing piece of work. Does that make sense, sir? Doesn't have to be always there, but many
Yeah, that's what I want to know because
yeah.
I work as in a public health. So if I say, for example, tobacco policy should be from my study, I understood the tobacco policies has a gap in this area. So in the future, you should. So what I'm writing is we should have a studies to develop a better policy. That's what you mean by
Yeah,
that.
yeah, you can say that. Or maybe in the future we plan to do this study in a, let's say, a bigger cohort, or maybe using something like that. If you plan, it is not always necessary. Sometimes
Okay,
in
fine.
more of
Thank you.
basic research, laboratory research, planning, original research papers. So because in public health, a lot of the studies that you are talking are mostly systematic reviews, right? You are not so typically future planning is for original research papers where you have primary research data.
Yes, now I got it.
Yeah. So I
Thank you.
think,
Thanks a lot.
Dr. Pati, uh, that would be all for the session, I guess. I am putting in my email ID. So if there are some questions, please feel free to write into us.
I just one question.
Yes, sir.
Ma'am, actually, there are journals who do not take payment and they say gold open access, but they have a policy that you do not have to share that article for 2 years or such. So, in that
Yeah,
case,
yeah. In that case, also, just look into Sherpa Romeo. When they say do not share the article for 2 years, so no, if they don't, if it is gold open access, then there is no embargo. You are talking about journals which probably have an embargo, and
yeah,
after that embargo, it becomes free, like British Medical
yeah,
Journal there and all that. But even in British Medical Journal, the final accepted version, not the version with the journal logo and everything, that cannot be shared. But the final, see, before you the journal, before your manuscript goes to proofing and galley proofing and actual publication in the journal format, you get a final accepted version, right? Okay,
yeah.
with the review where you have incorporated the reviewer comments, everything that, which is the final version for all purposes, for all scientific purposes. That version you can upload into your personal homepage
Okay.
or into
And which are the servers where you were telling we can
uh,
upload?
bioarchive is there. These are
Okay.
preprint repositories, but you can put your article and say
Okay.
this is a postprint.
Okay.
You just tell them this is not a preprint, it's a
Yeah.
postprint.
Yeah.
Yes.
Thank you.
Dr.
Thank you, friends. You can post your questions to the email. I hope this is a weekend and Dr. Nag already spent more than 1.5 hours. So, Smita ma'am, do you have a final question or final comments?
No, I just wanted to say, Dr. Divjot, you can do it on the research gate on all those.
Yeah, ResearchGate also. Yes.
We can post that post print in the research gate as well.
Yes.
Just try not to put the journal format. Don't take the PDF, final PDF
Yeah.
from the journal and post it, because that is when you are going to be, hitting the copyright violations.
Yeah.
But the final accepted version, you should not be having a
Okay.
problem.
Thank you.
So that is all from me today. As I said, you have my email address. Let us know if you have some more queries, and we'll be glad to help, answer them. And again, thank you, Dr. Pati, for the kind invitation, and wish you a lot of success in this national workshop that you are organizing.
Thank you, ma'am. And we also appreciate your within a short notice, you accepted our invitation. And in future, we will also like to avail your expertise in our forthcoming workshops and seminars. And friends, let us give a big clap or a thumbs up for Dr. Nag.
Thank you, everybody. Thank you. Yeah. Goodbye, and do have a great rest of the Sunday.
Yeah. Thank you so much. Thank you.